All right, welcome back. So, in this third video on the properties, structures, and functions of macromolecules, we're going to go over proteins. And just to make you aware, this is probably going to be um, the most difficult one, but also the most important, because proteins, specifically enzymes, are going to be very important on the test. So, I want to make sure that I have time to go through this clearly and that you have time to work on it. All right. I might suggest doing this first, spending the most time on this, and spending less on the other three, especially nucleic acids, because when we do genetics at the end of the quarter, we're going to be spending a lot of time with DNA and RNA. But that's neither here nor there for right now. Proteins are important because they're the most structurally in how they're shaped and functionally what they do diverse group. They've got so many different jobs. Here are just some of the functions that proteins fulfill. They're involved in almost everything, and I'm not going to read them because you can do that. But if you just look at that list, a lot of these things we're going to talk about more in future units. But the main takeaway is look at some examples here to remember and just know that they're involved in almost everything in a living organism. Proteins, like all other macromolecules except lipids, are polymers. The monomer, the repeating unit of a protein, is an amino acid, and we'll talk about the structure of an amino acid a little bit later. Here's an amino acid right here. Here's another amino acid. So just like all the other macromolecules, we build polymers by joining them via dehydration synthesis, removing the water to form polymers. In our bodies, there are 20 different amino acids that are used to build up our tissues. There's more amino acids than 20 that exist. There just two of them really aren't found in living organisms all that much. The polymer we call polymers of amino acids, a bunch of them linked together, polypeptides. So in linking these two monomers, we get a polypeptide chain made up of two. Sometimes we just call that a dipeptide. So one important thing right away, don't get a polypeptide confused with a protein. A protein is like the finished product that does some sort of job. So a protein can be made up of only one polypeptide chain, but they could also be made up of more bound and folded together. So a protein has more to do with, this grouping of polypeptides does this function. Okay? So a protein has a specific function. Polypeptides in one or in multiple copies make up a protein. Proteins are very large and complex molecules. Look at the ones on the bottom for biscohemoglobin and the human growth hormone. They have very large, complex 3D shapes, and that shape is very important. Just like a puzzle piece, that the shape of the puzzle piece tells you where it goes, the shape of the protein tells you what it can do, what its function is. So let's go all the way back. We took a big picture view all the way back to amino acids, which is the building block. This little drawing on the right, this is the general structure, the general structure of an amino acid. So remember, there's 20 of them. All 20 amino acids have these parts in common. They have a central carbon atom that has four things attached to it. On one side, it has a hydrogen atom attached to it. On the other side, we have a nitrogen and two hydrogens. We call this an amino group. That's where the amino part comes from, an amino acid. On the other side, the opposite side of the amino group, we have a carboxyl group. All right, COOH. All three of these things are exactly the same in all 20 amino acids. What changes or differentiates each amino acid from the other is this. In this picture, it's just labeled R, and we call it the R group or the side chain or the variable group. Different amino acids each have something different attached here. It could be one atom, it could be a grouping of many atoms. But this R group confers very unique chemical properties to each amino acid dependent on the properties of that R group. So if that R group would be, for example, hydrophobic, this amino acid would be a hydrophobic amino acid. Or if that R group was acidic, then this would be an acidic amino acid. So that R group is very important in determining the properties of each amino acid. And so we can use these amino acids and put them in different combinations, just like different combinations of letters and words, to make different polypeptides. So here is a picture of some. No, that's all. I just had to count. That's all. That's all 20 amino acids. And in blue, these blue boxes are each amino's R group. The things in black, that's all the same. That's like what we had here. That's this part. That's the part on top. These R groups are all different. Some are really big and complicated, like in tryptophan. Glycine's the simplest one because its R group is just a single hydrogen atom. And so you can see how they're very different and very diverse. And those differences give them important properties. All right. Now, there are two special amino acids, cysteine and methionine, that contain sulfur and can form disulfide bridges. And this is what a disulfide bridge is right there. So it's a covalent bond or cross-link between sulfurs that helps to stabilize the protein. So if we have maybe two cysteines facing each other, they could form a disulfide bridge, that little bond right there, to help stabilize our protein. So if we're going to build proteins, we're going to form what's called peptide bonds. That's what the bond in between one amino acid and the next is called. It's a covalent bond between the amine group of one amino acid. See, the amine group of one and the carboxyl group of the other. So when we remove H2O water from dehydration synthesis, we form a carbon to nitrogen bond, a carbon to nitrogen bond. That's what a peptide bond is, that carbon to nitrogen bond. Notice that when we're left, our dipeptide still has a carboxylic acid group or a carboxyl group, still has an amine group, so we could add another amino acid to either side and keep making it bigger. We refer to the two different ends of a polypeptide as the N-terminus, the side that has the amine group, this is the N-terminus, and the C-terminus, the side that has the carboxyl group. When we get a whole bunch of amino acids linked together in a big long polypeptide, you'll start to see a pattern in the chain. It goes NCC, 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 NCC. Okay? It will have that same pattern repeating and repeating and repeating for as far as it goes. NCC, NCC, NCC. All right. Now the last part I want to talk about is probably the most important, which is called protein structure and function. If we're going to remember one thing, it should be that the function of a protein depends on its structure, its 3D shape. Is it twisted? Is it foiled? Is it coiled? Is it in a unique shape? Is it flat? Is it round? Does it have a big, kind of divot out of it? Its function is determined by its structure. Look at the different proteins here. You'll notice they have very different structures. They also have very different functions. Collagen's like inner ear, it's structural. Pepsin is an enzyme in your stomach. Hemoglobin is in your blood. Very different functions, very different structures. Now, this is when I start to kind of lose people, and so I'm going to go kind of slow with this. There are we call it four different levels. There are 4 levels of structure. That should be an S, not a fancy F. There we go. And they have names. Primary, secondary, tertiary, and quaternary. They all in some way contribute to the final shape of your protein. The primary structure of a protein or of a polypeptide is the amino acid sequence in the peptide bonds. In what order are the amino acids linked together? Even if two different polypeptides use the same amino acids, let's say they use the same 5, but they put them in a different order, they would have a different structure. So covalent bonds occurring between the amino acids and what order they're covalently bound is the primary structure. And that information is ultimately determined by DNA, like we will see a little bit later. The secondary structure. The secondary structure looks like this. And there's 2 examples. There's a beta pleated sheet and an alpha helix. Secondary structure is caused by hydrogen bonding which occurs between the backbone of the polypeptide. So it doesn't involve the R groups at all, just the backbone. So no, it does not involve the R groups. It just involves the backbone. Look at here. Hydrogen bonds between oxygen and hydrogen in the backbone, making a helix or sheet, pleated sheets. Hydrogen bonds are important in secondary structure between the backbone, not the R groups. The third or tertiary structure is where R groups become really important. We talked about how all the R groups have different properties: hydrophobic, hydrophilic, ionic, acidic, basic. Those properties will cause the polypeptide to bend in certain ways. For example, If here, let me draw out on the side, if here is a polypeptide chain, and here is one R group, here's another R group, it's a third R group, it's a fourth R group, and pretend the ones I'm putting circles around are R groups that are all hydrophobic. They do not like the water. They want to get as far away from the water as they can. And so it will cause them to cluster together on the inside of our polypeptide. And in so doing, our polypeptide chain will bend and scrunch up together like this. There's another one over here. Changing its shape. So the interactions, the R group interactions, how their properties cause the polypeptide chain to bend, contributes to the protein's tertiary structure. And lastly, we have the quaternary structure. Now, all proteins don't have quaternary structure. Like I said, some proteins are only made up of a single polypeptide chain. In that case, they do not have a quaternary structure. Only proteins made up of multiple polypeptides, have a quaternary structure. And the quaternary structure is what way do the different polypeptides fit together? Which way are they oriented together? There are many different ways they can be oriented together. What is the way that they are oriented together, fit together to make a big overall structure? Right? So maybe pause and make sure that you can understand this image, what I'm talking about. If you have some questions, I'll answer them probably on our next synchronous session with this. All right. Lastly, proteins can denature, which means that they can unfold. Like when we saw here in the tertiary structure, a protein chain all folded up. When a protein denatures, it means it unfolds. All right, if you've ever, for example, fried an egg on a stove, and it goes from being very clear and liquidy to very white and rubbery, that's denaturing. Right? And what denaturing does is it disrupts the hydrogen bonds, the ionic bonds, and the disulfide bridges that stabilize the protein's tertiary structure. It breaks those bonds, it adds energy via heat or some other mechanism, it breaks them and unfolds them, just like in this picture. Unfolds, denatures. And so if a protein denatures, it's not the same shape. And like we said at the very beginning, shape matches function, and therefore it can no longer do its job again. Some proteins can return to their functional shape after denaturing, but many can't. You might have heard of a recent experiment scientists did that tried to unfry an egg. Unfry an egg. That was basically what they were doing, is looking for a way to undo the denaturing process. So, temperatures, extreme temperatures, or pH or salinity can cause proteins to denature. Different proteins have optimal temperatures and pH and salinity based on where they're found in the body, and departing from those specific temperatures or pHs or salinities will cause them to denature. So, for example, amylase, an enzyme in your mouth, a protein, an enzyme in your mouth that helps to break down starches, has a very specific temperature and pH that it works best at. Now, peptidase that works in your stomach has a much different optimal pH than amylase. And either one, if you remove them from that environment and change the pH very much, they will denature. So denaturing alters the secondary and more importantly the tertiary structure of a protein, alters its 3D shape. All right, so that is the end of proteins. We are going to spend some time in our synchronous session on this because it usually is what students have the hardest time with in this unit, but it's also the most important. So send me questions or log on to the discussion in Canvas. Otherwise, we'll see you at that time.